This video is on how to measure reaction rates through experimental techniques. So there are 6 different ways I'm going to briefly talk about. Make sure you read in the book about these and look at some of the diagrams and pictures as well to more thoroughly understand this. So the first one would be a change in volume of gases produced. So if you have a gas that is a product, like CO2, you can measure this by, I've got a picture right here, this gas syringe here. So if you had the gas being produced from your flask, let's say, and you had a tubing hooked up to a syringe here, the syringe would expand, right? It would go this way as the gas filled it, and you could actually measure how much gas was in there. Another way to do it would be an inverted burette, like in this picture. We actually did something similar to this with our molar mass of butane lab, where we had the butane lighter underneath in the bottom here. And as the gas filled that, we could measure the volume of the gas that way. And lastly, another way to do it would be data loggers. And these are with those lab quests we've used in class. There are different probes and sensors that you can hook up to them that will actually measure the volume of the gas that is released. So a second way to do this would be to look at the change in mass of a gas. So also, if it produces a gas, you can measure it on a balance and see how much the mass changes. So let's say you had a reaction happening in a flask or a beaker, and you set it on the balance, so you have an initial mass of everything, and then as it reacts, the gas leaves and the mass should decrease. One thing this wouldn't work for, though, is if you had hydrogen gas, because hydrogen is too light to notice any obvious change in mass, especially with any of the balances we have. So a third, going back down. A third method would be to look for a change in transmission of light. So this is called colorimetry or spectrophotometry. So what we're looking for here is, do we have reactants or products that are colored? Do we have a color change? And what you're going to notice is, this will be when you have transition metals. So you're going to look for transition metals in your reaction. So typically they're either going to be alone on one side and a compound on the other side, or you're going to see them with a change in charge. So you'll see there's different absorptions because there's different wavelengths of light as the color changes. Sometimes an indicator is added. Let's see, we use a colorimeter or a spectrophotometer to measure the intensity of the light. And lots of times this could also be graphed in absorbance versus time. And that's where some of those data loggers could also come in and help with that. The fourth method would be a change in concentration using a titration. And we talked about titrations briefly before, and we're going to more thoroughly when we get to acids and bases. But remember that this is changing the concentration of an unknown and comparing it to a known standard. So you have one in your burette that you know the concentration of, and a different one in your flask down below. And as you add the known standard, an indicator will usually tell you due to a pH change. So what we're looking for here is, what we can do is we can't do this continually. So you have to do this at certain times at regular intervals. And here's where like a pH meter would come in handy, because you could have it record every, 2 seconds or things like that. There is a process called quenching, which can actually like freeze the reaction so it doesn't continue to change. But it is very difficult and very time consuming. It's like that is not something we would do here in our lab. What you're looking for in this situation is, because you'll have a change in pH, and I know we haven't talked about that really yet. So to see that, you'll see either a hydrogen ion change, like a hydrogen ion decrease, or a hydroxide ion increase, because hydrogen ions are related to acids, hydroxide are your bases. All right, the fifth method is a change in concentration measured due to conductivity. So this has to do with ions and an electrical charge. So what we're looking for here is a change in ions, more specifically in the number of ions. that you'll see on like the reactants and product side of a reaction. If you were doing this experimentally in the lab, here's where you'd use a conductivity to actually measure how much electricity was conducted. All right, and then lastly, and this is the one we're actually going to do in lab, is what we call a clock reaction. So we will actually measure the time it takes to reach what we call an endpoint. So like when the reaction is done. So we will, for example, time reactions to see when they change, how long till they change color. And we will manipulate them differently. So we'll change different factors related to that reaction to see how that affects how long it takes for the endpoint to appear. So make sure you read about this more in the book, but these are the 6 experimental techniques that you need to know for measuring rates of reaction.